from this cartoon, I guess, I just want to show that I'm working on analyzing experimental data using either theories or hypotheses or computer simulations. So in our lab, one of the data that we analyzed was showing actually yesterday by Sunny was that when the proteins bind to DNA, there was allosteric effect. That means one protein binds affects the second protein binding. And there actually we found there's some kind of DNA sequence dependent, particularly depends on the base steps, whether it's CG rich or is TP rich. So that also echoes to the first three talks we just heard because CPGs are prone to methylation. So then we try to analyze from a chemical view why they are different. So for example, if we look at here, you have APT and TPA. They should not be much different in terms of chemical compositions. But in terms of base pair stacking, you can see that there's a very large difference. If A on top of T, then you will see the two base pairs, they overlap very well. If T on top of P, you see that overlap is almost gone. That can be seen from this overlapping area we project onto the z-axis. You will see if it's TPA, you see poly AT, you will see two peaks. The small overlapping is for TPA, the large one is for APT. But if you have CPG, you see that this is more or less the same, whether it's CPG or GPC, the difference is much smaller. On the other hand, if you methylate the C, you will see that the blue curves, in terms of stacking, actually now methylated CPG behaves somehow like TPA. So that prompts us to just look at how these dinucleotides are distributed in different genomes. It was very interesting if you look at the bacterial genome, for example. For CPG, there is a very sharp valley near the transcription start site. One might see because this CPG density is very high for that species. If you look at the different species, for example, bacteria living either in acidic conditions or high temperature, they tend to have a low CPG. Even their CPG density is already very low, lower than expected, than random. You still see at the TSS there is a sharp valley at there. So along the evolution, actually, you will see the signal begins to change starting from a valley all the way to changes to a peak. So finally becomes something like the CG island. So CG island is well known. That probably tells us the composition of the dinucleotides already has something to do with the gene regulation mechanism. So what we do is we just take the dinucleotide distribution around the promoters and the gene bodies and to see whether we can classify different genes. In fact, we can easily separate the genes into three classes. The first class, you can see that they are really very rich in housekeeping genes, and the second class actually is mainly developmental related and neural related genes. And third class, shown in blue here, they are really somatic tissue specific genes mainly. If you look at their CPG density, you can see that the first class has very high peak, very pronounced near the TSS at the promoter, and the relatively low CPG density on the gene bodies. The second type, which is developmental or neural-related genes, they also have a peak at the promoter, but a very high CPG density on the gene bodies relative to the other classes. The third class actually has very low CPG density all the way. So there are many properties actually showing such behavior. We heard about G quadruplex yesterday. And if you look at, for example, the G4 distribution, it also actually follows the same trend in the three classes. So then one might say, maybe they will affect not only the other properties, such as the chromatin state. So we can draw either aminase, DNase, or attack data and align them along the transcription start site. You will see the class 1 genes actually, they show a very clear open chromatin structure near the TSS promoter. And then on the gene bodies, transcribed the gene bodies, you will see that very periodic peaks. At class 2 genes, this feature is blurred, and class 3 genes, this feature is almost gone. So then we can take the periodicity and analyze it. One finds actually the nucleosomes for the class 1 gene, they are organized with a periodicity about 167 base pairs. So 147 plus 20. But for the class 3 gene, which are normally silent, and their actually periodicity is about 181. So that's longer, actually. The distance between two nucleosomes in terms of the DNA length is larger. So because we know that methylation is somehow related to the chromosome positioning, so we also take the methylation and do the same analysis. And we find exactly that for the class 1 genes, it shows the periodicity of a methylation peak of 167, and class 3 is 181. So these two data agree with each other. One question is, what happens? Why? It looks like euchromatin are even have a higher density of nucleosomes. So if you look at the structure and analyze it, one finds that the class 3 genes actually bind the H1 linker stronger. And the class 1 gene actually does not bind H1 histone linker strong enough. In fact, one can analyze if you have TPA-rich sequences, they tend to interact strongly with H1, but you have CPG, then actually they interact relatively weak with H1. So we know the CPG density distribution is not even, but not only at just one scale. Actually, they are arranged in different scales. First, the CPG density is lower than expected for human genome, and it's uneven distributed to form a CPG island. And if we further analyze, if you draw each of the CPG island as a line here, you will see that CPG islands also distribute very unevenly. And we call this forests and prairies, and then if you analyze the CPG density actually of many different scales using a wavelet analysis, you will see that the CPG density is distribution is uneven at all these scales, but it's not random. Actually, this distribution has a tree form. So that tells us it's a really hierarchical arrangement of the CPG. So we know that that's not very rare, because we know from the compartment analysis from Hi-C data, we know the chromosome structure or chromatin structure is also hierarchical. You take the compartments of those, each of the cell, you can take the same wavelet analysis. You will again see this hierarchical distribution. Actually, the CPG density distribution and the Hi-C map, they correspond each other very well. Even if you look at the expression, you will see this hierarchical distribution, and they all actually resemble each other a little bit. So that tells us probably the CPG density distribution, especially those large domains, which one is rich in CG island, the other is poor in CG island, actually they really tend to segregate in 3D structure. So we take the Hi-C data, then reconstruct the model. You will see the blue, the so-called P domains, they tend to segregate into heterochromatins, and the red domains, the F domains, tend to segregate in 3D space in euchromatin. Actually, the super resolution imaging data show the same segregation. And the interesting genes are those in the blue domains. They tend to be tissue specific. And only in certain tissues or certain cell states, actually, they are mixed into the red. And once into the mix in the red, actually, they are upregulated, and they are tissue-specific in terms of function. So just look at which blue genes are in the red domain. You can tell what cells they have. And the boundaries between the 1D sequence property, the F and P, overlap very well with the compartment and the TAD domain boundaries. But if you look at just the CG Shaw, which was another popular way of dividing the human genome, that overlap is much poorer. So we also said that probably earlier said we found that there's some correspondence between DNA methylation and the chromosome, the nucleosome positioning. So whether the DNA methylation will have something to do with this 3D structure or the 1D sequence, you will see that by this analysis, one sees those silent domains, they tend to be demethylated, or the methylation level is lower. Those are the open Cs, not the CG islands, because we think the open Cs reflect better the global structure. And the forest domains, which are the red domains, actually, they tend to be with a higher methylation level. The interesting methylations actually occur to exactly those blue genes. You can see this, take this example. This gene has very low CpG density on the promoter, actually just 1 or 2 CpGs. And if it's in the blue domain, it's in the heterochromatin, its methylation level is indeed very low, very high. But when it's upregulated, that's in liver, actually you will see that this even one single site of CpG somehow knows how to be demethylated. So one can have many, many properties certainly associated with the CpG density. One of the examples we heard about is the repeating sequences. So the repeating sequences, just line and sign, they also tend to segregate. But if you get rid of the influence of CpG, you will see that they do not segregate as apparently. But if you get rid of the distribution influence of line and sign on CpG, you will see the CpGs, the segregation according to CpG still exists. So that leads us to think probably actually the dinucleotide distribution has something more to say than the repeating sequence in terms of the chromatin structure organization. So let's look at the chromatin structure organization. One of the problems with Hi-C is because it's highly noisy and sometimes misses the very significant interactions. So we need to develop a mathematical tool to denoise at the same time to enhance the signaling. So this is the method we developed. We call it CTG. I won't go into the mathematical details, but I just want to show that if you have just Hi-C, if you compare to super resolution imaging, you see there's still difference. But if you now using our method to treat the Hi-C data, you get CTG matrix as we call it. You see there's a very good correspondence between the CTG matrix and the Hi-C, the resolution, high resolution imaging data. And in terms of distance, actually, there's also very good correlation. The method is designed to treat sparse data, so it works equally well for ChIA-PET, single-cell Hi-C, and micro-C, even single-cell micro-C data. So with this method, then we decided to look at samples from cancers and to see whether this methylation pattern has something to do with the chromatin organization. So we take the basically mini bulk, and Yan Yi basically did most of the experiments there. So here I show a few examples. For example, for the oral cancer, there's OSCC and ADCC two types. And if you now analyze the chromatin structure, you will see that they tend to change from normal to cancer. They tend to change in the similar patterns. And overall is the loss of the long-range contacts and the gain of short-range interactions, in particular, the interactions between adjacent TADs. And that tells us probably there's general property of the structure changing when one goes from normal to cancer. So one can take each of the points to train a model to see whether that, just look at each gene, how it interacts with other genes, whether you can see from that point, whether you can tell this is from a cancer sample or from a normal sample. If the chromatin structure is important, one should be able to tell the difference. Indeed, we can see that the successful rate is very high. For any single point, you take it out, you can predict with 90% of accuracy that whether it's normal or it's cancer. You combine all these points together, you will see there's very big difference between normal tissues and cancer tissues. And also there's a difference between normal tissue and normal tissue taken from the cancer patients, actually. So using that method, then one can easily classify the different types of cells. For example, the two types of oral cancers and also their adjacent tissues. In fact, that can also be used to classify, to our surprise, two types of ADCC. One is solid, the other is cribriform. So these two actually, you can see from the samples that they look different, but the molecular difference is largely unknown. That tells us their chromatin structures are already different. So maybe that chromatin structure has something to do with something else as well. So we know that in cancer, what also important is copy number variation. So we look at the copy number variation and found actually surprisingly for many different cancer types, although their copy number variation number can be very different, but their patterns are very, very similar, just like showing here. And when you compare to what's going on is that the copy number variation changes occur largely at the edges or the boundaries in the high C. So those are the isolated domains that they tend to have copy number variations. So you can see this is the basically the statistics, and these are four examples. You have the first one that's a normal normal tissue. You will see that high C is have a lot of long-range interactions, and the second, the third are from cancer patient. The second one still have a lot of long-range contacts in terms of chromatin structure. The third one, actually, the sample lost much of the contact for the lower corner. So part of the DNA actually is losing contacts with the rest of the DNA. But there is no CNV change for these patients. The last patient actually has this both copy number variation and high C change. So there's basically a correspondence between the third, fourth, tells us probably the chromatin structure actually can change in a way that facilitates the CNV change. And the next thing one can look at is the fusion between different genes. You will find that the genes that tend to fuse together tend to have close contact in the chromatin. Actually, not only in the cancer cells, in the normal cells, they were already in close contact. Now, if we look at the methylation change, again, most of the forest domains in the compartment A, they remain highly methylated in the open C, and the prairie domains actually began to lose much of the open C methylation. And here shows the difference. So in cancer, the solid lines, the difference between the forest and prairie domains, they get enlarged. And from normal to stage 2, 3, 4, you see there's an increase of the difference of methylation. Not only for oral cancers, for other cancers, you find similar things. And what happens is that actually the methylation occurs seems already at stage 1. So you can see that 1, 2, 3, 4, they all have increased hypermethylation of CDI and hypomethylation of the open C. In fact, if you look at the expression also changes already from the normal cell already at the first stage. So those genes that are highly expressed in the fourth stage, stage 4, already highly expressed in stage 1, those highly expressed in stage 1, they remain highly expressed in stage 4. And those is similar for demethylated ones. So if everything ends up here, then I will feel very happy. There seems to be correspondence between methylation and structure. But seems biology always gives us surprise. There's this ADCC we talked about. If you look at its methylation, actually it does not show strong hypermethylation and does not show hypomethylation. In fact, if you compare OSCC and ADCC, you will see its methylation level does not change in cancer. And for the OSCC, those that get hypermethylated as known from the H3 K27 methylation, trimethylation marked regions. But actually, such a pattern is also not seen in ADCC. In fact, this picture makes it even more clear. If we look at the difference between gland and ADCC, at multiscales, there's no much change from normal to cancer. But for OSCC, certainly there's a very dramatic change in terms of methylation pattern. And this methylation pattern now actually resembles quite similar to the CpG density distribution again. So at multi-scales. So finally, we analyze what really, at least for these two cancer types of cancers, what really correspond or correlate with the expression change. Indeed, the compartment does correlate, and methylation correlates very weakly. And if for OSCC, because it has a lot CNVs, actually they correlate also relatively strongly with expression change. So with that, I want to give a very quick summary. I think the dinucleotides are distributed unevenly is seen in many, many different genomes, and actually shows evolutionary trend, which I did not have time to show. But the effects of these dinucleotides and distribution actually can be seen in the mechanical properties as well as biological functions, it seems, or at least they are correlated. Starting from single base pair to compartments and even actually to territory. So demethylate changes in cancer development are closely related to multiscale CpG density and chromatin structure. Also, we think there's rich information that is buried in the 3D chromosome structure, not only just one scale, but in all these scales and their interconnection. But it's really complicated and yet to be deciphered. At last, actually, just want to say that I said earlier in the talk that you will see sharp transition of methylation chains from normal to cancer different stages. And for example, for the low-grade tumor, for esophagus, the change in the methylation is very, very weak. But in fact, you already see in the chromatin structure, there is breaking of TAD boundaries. You began to see that different genes from different TADs began to interact. And that trend gets stronger when one gets into lower stage, higher stage of the cancer. So probably there's something buried in the structure itself. Okay, thank you very much. That's my